Thank you so much, and welcome to that session. It's an honor to be allowed to give that summary, because there's so much going on. It's small steps, but there are steps in the right direction. So let's start. I do this on behalf of the Scientific Advisory Board of the ULF. My disclosures. So the first talk was given by Marjo van der Knaap. She has founded the Amsterdam Leukodystrophy Center and has been working on leukodystrophies for almost 40 years now. And she identified a new leukodystrophy, and that's a leukodystrophy with a quite straightforward treatment, which makes it, of course, very interesting. She had 4 patients, 2 of whom were seen at our center and are therefore documented in detail. Patients had a big head. You see here the growing head circumference. They had also some episodes of increased neurological symptoms and also signs of elevated intracranial pressure like vomiting and drowsiness. These patients were treated with corticosteroids for these episodes. All patients also had cognitive problems, autism, didn't develop speech, and had a severe motor disability, mainly ataxia. And when you look at the MRI scan, you see at the age of 1 year a relatively normal MRI scan. But when you look at the brain cortex, that looks a little bit thick, a little bit voluminous. And when you see that child at the age of about 5 years old, you start seeing a lot of white matter abnormalities, and the white matter also gets a kind of a swollen aspect. Genetics was actually quite difficult, because the first genetic testing and the second genetic testing didn't show any diagnosis. But when the DNA results of the children were reviewed, they found in all children the same heterozygous variant in that gene, SCL4A4, and that gene encodes a transporter for sodium. So that is important for the exchange of sodium from the cell into the extracellular volume. And it was difficult to find that mutation, not because we didn't see it on the genetic sequencing, but because the disease associated with this gene was an autosomal recessive disease, so that needed 2 variants on both alleles, one from father, one from the mother. And the difference between that classic picture and these patients was that these patients only had 1 variant. So that was filtered out by the genetic techniques, because one thought you need to have 2 variants to get a disease. But that specific variant actually is enough in itself to lead to a brain disease, not to a kidney disease. When they looked at functional studies, you see that this channel, which actually should control the sodium transport between the cellular space and the extracellular space, it's open, so the sodium can leak out of the cell. And when you have that, actually, for example, in the kidney disease associated with this gene, you give a bicarbonate or baking powder, which actually helps to get the sodium concentration more in equilibrium. So they did that for these children, and what you see is that the white matter abnormalities greatly improve, and there are also some clinical improvements. So children have a better mood, learn new things, and you see that the head circumference stabilizes with the treatment. So this is a very rare disease, but a disease which is at least partially treatable. Do you have any questions for that talk? If yes, there are microphones and you can get up to ask your question. No? Okay, so let's move to the next. Yes?
Just a quick question. How was the sodium bicarbonate administered?
Oh, orally. Yeah.
Okay, thank you.
So it's a relatively easy treatment. The next talk by Davide Tonduti from Italy, Milano, was about prognostic factors in Alexander disease. And the problem in many leukodystrophies is that we don't know at the beginning of the disease how severe the disease is. So when a baby has the first symptoms of Alexander disease, it's not always possible to predict how severe the disease will be. You have, of course, some information of genetics, but as you see that many forms of Alexander disease are possible with the same mutations. So you have this classification of Alexander disease, and the most severe one means that you don't develop any motor milestone, so no head control, for example. That's important, that information, of course, first of all to counsel parents, but second also, if you want to do a trial in Alexander disease, you would like to put the same group of children in a trial cohort to be very sure of effects, possible treatment effects of a tested drug. So that's one of the reasons why we are so interested in being able to predict disease severity at a very early age. And with a big collaborative study involving people from different continents, together with information of the MRI and head control, yes or no, you can actually predict relatively well the clinical severity of Alexander disease. So this is an important milestone for this disease, and that might also be a possibility for other similar diseases starting very, very early in life. Any questions for this talk? No. So the next talk is from Amsterdam, from Daphne Schoenmakers. She just finished her PhD on metachromatic leukodystrophy, and she looked at the effects of allogenic hematopoietic stem cell transplantation in late onset metachromatic leukodystrophy. Late onset metachromatic leukodystrophy means late juvenile and adult onset patients. Late juvenile starts from the age of 7 years old and adult MLD from the age of 16. And this is a group for whom a gene therapy is not registered. So we treat those patients at the moment still with this hematopoietic stem cell transplantation. And it's also a group where patients have very different disease severities. First of all, age of onset can be very variable from age of 7 or 8 years until the age of 40 years, or even a little bit older. And then you have certain variants in the gene for metachromatic leukodystrophy, which give you a very stable disease course motor-wise. The other challenging problem is for that study that in many centers only very few or even no patients with adult forms of metachromatic leukodystrophy were treated, criteria to treat or not to treat patients were very diverse, and follow-up was often very basic, meaning that we had to do a lot of detective work to find out how patients are doing after this transplantation. But Daphne managed to do that, and it was actually a quite surprising finding for us. You see that patients who were treated with that treatment have less severe disease manifestations than untreated patients. You see here many more dots than in this group of patients. What you also see is that treated patients have much less evident cognitive impairment than untreated patients, but you also see that if you treat symptomatic patients, you don't see any difference with the untreated patients. So you do need to treat early on if you want to see treatment effects, positive treatment effects. What was also quite surprising is, and I think we miss a part of the slide, is that even though patients were treated, patients do develop, in most cases, progressive disease. The blue group is patients with progressive disease. And that is something which is perhaps not so nice to hear, but it's important for us because even if we get newborn screening in the next 2 years, there will be still a large group of adult patients or late onset patients who are not picked up by a newborn screening because they are already born. And we need to know how that treatment works if we want to counsel families and patients well. So for us, it means also that we need to change our expectations for the treatment. It's not a cure. So even patients treated pre-symptomatically usually are not able to work in a normal work position and have all kind of more or less severe problems. And that's not a very positive result perhaps, but it's good to know. And it makes it even more urgent to diagnose those patients early. And perhaps also to advise in cases like those symptomatic patients that it's better not to do the treatment, even if you would love to offer something to families and patients. Any questions? No, also not from the chat. So the next talk was a very basic talk about Krabbe disease or globoid cell leukodystrophy, as it's also called, by Daniel Segal from Tel Aviv University. In Krabbe disease, you have accumulation of certain molecules which are called galactosylceramide and psychosine. And those molecules accumulate in cells and are toxic for cells and lead to demyelination, loss of myelin. So Dr. Segal found out that when you have accumulation of these substances, they organize themselves to form like fibrils, like fine structures, long, thin structures. And these accumulation of proteins are toxic to cells. And the question is whether these accumulated substances, galactosylceramides and psychosine, are perhaps less toxic if they don't accumulate to those fibrils. So this is something to look at. They tested certain compounds which could inhibit this fibril formation in a cell model and in a test tube. So of course it's important now as next steps to look whether these fibrils are really also present in a Krabbe disease patient tissue, especially brain tissue, and whether these compounds can stop the formation of fibrils and also help preserving cells. So this is something which will take some time because you first would like to see in cell cultures and then in the mouse model. And then you have to see whether those compounds could be also given to patients with Krabbe disease. But it's a very interesting approach to look at the behavior of storage material, because usually we think we have storage material and that accumulates and the cell doesn't like that. But this is something extra where you really look at what do those storage materials do they organize themselves or not. So I don't see any questions. You have one?
Great, so these are both from online. They're both actually going back to your last slide in MLD and come
Yes.
from a doctor? Yes. How were presynaptic late juvenile patients identified? If no symptoms were present, what was it based on?
Oh, usually it's brothers or sisters of index patients who have not yet developed clinical symptoms.
Okay, fantastic. What are your thoughts about using LibMeldy for late juvenile, given that stem cell transplants without gene therapy is not very good? Sorry, do you want me to repeat that?
Yeah.
What are your thoughts about using LibMeldy for late juvenile onset, given that HSCT without gene therapy is not particularly good?
Yeah. That's an obvious question. We also asked ourselves, could LibMeldy be better than the allogenic hematopoietic stem cell transplantation? To be honest, I think it will be very difficult to have a good trial. So we might not be able to answer that question scientifically. But I also would say that we can extrapolate the findings from the late infantile and early juvenile cases where we know that gene therapy is better, really better than allogenic hematopoietic stem cell transplantation. And I hope that this, together with these data, actually will be enough to convince regulatory agencies to reimburse this treatment. But that's I don't think we will get better data on the late onset patients than these ones. So the next talk was given by Alexandra Chaplot from the lab of Dr. Bernard in McGill, Montreal. And she's almost defending her PhD on this subject. This is about an extremely rare leukodystrophy with hypomyelination, which is caused by variants in a gene called EPRS1. And that gene encodes an enzyme which actually is very important for protein synthesis because it connects the amino acid, which is a building block of the protein, to a kind of a transfer molecule, which is then needed to put all those building blocks together for synthesizing proteins. And when you have variants in this gene, you have a hypomyelinating leukodystrophy. So in 2018, some time ago, there were 4 children known with variants in this gene. And hypomyelination means that you don't make enough myelin in the brain. And it was not really known how mutations in EPRS1 lead to this myelin problem. So this group decided to study the clinical spectrum, but also possible mechanisms how this disease develops. And in a big work, they identified really quite a lot for such a rare disease of patients all over the world with mutations in this gene. And you see what we also know from other RS1 disorders, that you can have patients who do have a leukodystrophy in the typical clinical picture, but you also have patients who are even more severely ill with severe epilepsy, and other patients who don't have MRI abnormalities and perhaps relatively mild cognitive intellectual disability. When you look at the protein, the EPRS1 protein, it's important for two amino acids, and the proline part is the most important for this leukodystrophy. So almost all variants causing a leukodystrophy are in the part which is important for the amino acid proline. And we see that if you have mutations, your amino acid synthesis to that transfer molecule doesn't work well. And when that happens, you can actually make myelin-producing cells from patient cells, and they did that, and they looked how these cells could differentiate, and especially differentiate into oligodendrocytes. Those are the cells which are responsible for making the myelin in the brain. And those oligodendrocytes were not able to produce myelin, and what's also important, were very susceptible for stress and cell death. So this is important to understand how this disease develops in the brain, and that's also important for thinking of how we could influence this disease with new therapies. I think it's especially important to know also about the clinical spectrum in this disease. I see a question.
Okay, so in the brain we got the oligodendrocytes, and in the periphery we have the Schwann cells.
Yeah.
Is it possible that the same mechanism would be also affecting the Schwann cells?
Yeah, thank you for that question. We have quite a few hypomyelinating diseases where we know that only the myelin in the central nervous system is affected and not in the Schwann cells. The RS1 story is very interesting because you can have dominant mutations, and then you get a neuropathy, a peripheral neuropathy, which is usually which is not always demyelinating, but many of these patients don't have a neuropathy, so the peripheral myelin is normal. And why that is also a very interesting question, of course. Okay. The next talk also covered a hypomyelinating disease, POLR-related disease, or 4H leukodystrophy. That's perhaps the name we know this disease, and that disease is caused by mutations in different genes, all coding for subunits of what we call an RNA polymerase, and that's polymerase 3. And with this disease, we still don't have a good idea of what exactly goes wrong, why your oligodendrocytes are not able to produce so much myelin. And the question was, and that was actually looked at in the mouse model mainly, how do the mutations cause hypomyelination and neurodegeneration? And what this group found was that the transfer RNA levels And you might remember that from the previous slides, in all cell tissues were lowered. So that seems to be a problem when you have 4H leukodystrophy. And neurons and other brain cells are especially vulnerable to these low tRNA levels. And in mice, this not only leads to less myelin, but also to a chronic stress response, because cells don't like that, inflammation and death. And that may also explain why some patients in 4H leukodystrophy have this deterioration with infections, because your stress response is not working normally. But still, we have a quite long way to go with this disease to better understand it. So the next talk was about mutations in, a question.
Is the PNS affected by polar leuko, or is it isolated to just the CNS?
Mm-hmm. Yeah, in most patients we don't see a problem with the peripheral nervous system. That's actually a hypomyelinating disease where we do see a prominent peripheral nervous system involvement. That's also hypomyelination, and the disease is called Pelletier-Merzbacher-like disease. It looks a bit, if you look superficially at it, like as Pelletier-Merzbacher disease. It's autosomal recessive and caused by variants in a gene we call GJC2. And that encodes a protein which is called connexin 47. And connexins are proteins connecting cells. That's also why they are called connexin. And those are like a channel protein in one cell membrane and a channel protein in a cell membrane of another cell. They go together, so cells can directly transfer things they need from one cell to the other cell. So it's a bit of a circumventing all those fancy transporters controlling access and into a cell. And that these connexins also mean that you can make a big group of cells helping each other when they need, for example, to buffer potassium. It's not only going to one cell, but it can spread easily to other cells. So that means that cells can fix or can quite efficiently deal with a lot of potentially problematic small ions, like potassium, for example. And when you have mutations in that gene, you get Pelletier-Merzbacher-like disease, but with a prominent peripheral neuropathy, and many children also have epilepsy. And you again have mild forms and severe forms. In mild forms, you have spasticity at a later age. Perhaps you could compare that to AMN. And the question of this group in Chicago, Charles Abrams, was to understand why different mutations cause different severity of the disease. And they made a really nice molecular model. Unfortunately, the video doesn't work in the slide I received from the group. But when you look at that channel, here you can have the part of the protein which opens or closes the channel. And when it moves, it looks like small legs moving all the time. And that's quite interesting. Because we now can see exactly what mutations do with this channel, and that might also help in identifying molecules blocking or improving those movements of the little legs of this channel. Any questions? No? Okay. Okay, that is a very relevant talk for probably quite a few families without a genetic diagnosis of a leukodystrophy or other severe neurological diseases by Tomi Parkkinen from Kansas, and he looked at a new method to better sequence DNA. And when we think of DNA sequencing, whole exome or whole genome sequencing, we think that reading these DNA sequences is relatively straightforward, and we get a faultless transcription of a genetic code of patients and can find those little mistakes in the genetic code very easily. But for some mutations, that's not true, and they are so hidden or so difficult to bring forward by our usual methods that we know we have children with a certain diagnosis where we don't find a variant because our reading system, the sequencing, is not good enough. So this group tested a new method of genome sequencing, which is called high fidelity genome sequencing, and they showed that with this technique they could increase diagnostic rate in children, so giving diagnosis to previously undiagnosed patients. And that will be offered to a lot of patients with unsolved diseases, among them also leukodystrophies. Any questions? No? The next talk was by Josh Bonkowski, also from Kansas, and that was looking at death rates in patients with leukodystrophies, but also at the question, do we miss a lot of patients with leukodystrophies or not? And again, these are very relevant questions because as you all know, probably better than I do, leukodystrophies are a very underrepresented group of diseases when you compare it with, for example, childhood cancer. But probably death of neurologic diseases, including the leukodystrophies, is nowadays at least as high, perhaps even higher as children with cancer diseases, because our cancer treatment has become really good for many forms of childhood cancer, very much in contrast to the diseases we are working with. So they looked at death rates in hospitals and saw that, for example, female patients had a higher death rate than male patients. Not sure why that is, and they were working on that. And the other thing is that there was not much difference, no significant difference between patients with different racial backgrounds. The other question was, do we miss patients? So if you look at a lot of genomic data from patients with different diseases, and then when you find a mutation in a leukodystrophy gene, you go back and see whether that patient has been diagnosed with a leukodystrophy or not, or with another disease, you would find the missing patients. But even in the adult population, where we always think we might miss some patients because they are not diagnosed, there were no missing healthy or non-symptomatic adult leukodystrophy patients, with actually two exceptions. One is the ALSP, where we know that you can have the disease variant, but you either don't develop the disease in rare cases or develop the disease at a late age. And the other gene is SNORD118, which causes a leukodystrophy with calcification and cysts, therefore the name LCC. That's a inflammatory vascular leukodystrophy, and there were some patients who had a kidney disease, but no neurological disease. So that might be a group of patients, a small group of patients, not represented yet. I see a question.
Thanks. I don't know if I fully understand. How do you define death rate?
Yeah.
So we all die, so
Yes.
it's 100% death rate. So how would they define death rate in the study? Thank you.
Yes. That's a very valid question, because when I listened to the talk, I had exactly the same question in my mind. But the question is, of course, early death. And especially pediatric death, death in children, which is not the normal thing.
So you said that there wasn't a known reason why there was more females than males that died, but do you guys have a known ratio between male and females that have the leukodystrophies to come up with that statement?
Thank you. It's not. They have not a good explanation so far why they saw this difference. Of course, we know that some leukodystrophies are seen exclusively in males, so you could have even hypothesized that there are more males dying from a leukodystrophy than females. That would have been more understandable. But one of the possibilities might be a method, because they looked only at death in hospitals, not at death at home. So that's the next step, to see whether they can expand the study to all death in leukodystrophy patients. Okay, any other questions? No. Okay, the next talk was by Adeline van der Ver from a shop in Philadelphia. And it covered biomarkers, diagnostic biomarkers in Aicardi-Goutieres syndrome. That's a leukodystrophy caused by mutations in many different genes and characterized by an increased inflammation. So the body basically thinks it has to fight all the time kind of infections and get this inflammatory response is very high. And you can measure that with the interferon signature or interferon-associated genes, because measuring interferon, which is an inflammatory marker, directly is not possible well enough. And you can measure these interferon signatures in two ways. You can look at white blood cells and the markers they express on their surface, but you can also look at the expression of certain proteins by looking at RNA concentrations of these proteins. The latter test is not commercially available, so it's a kind of a research test in some hospitals. The first test, where you look at the cell surface proteins, that you can actually do on a commercial basis. But her group developed an RNA-based test, and she thinks that will be available commercially perhaps next year. Why is that important? The affected pathway in Aicardi-Goutieres syndrome, that interferon-producing pathway, can be modified by several kind of drugs. You can block the interferon A production, you can block the interferon A itself, you can block the receptor, and you can block the signaling when the interferon A has docked at the receptor. And there are several drugs doing especially blocking the receptor and blocking the signaling. And these drugs are already given to patients with Aicardi-Goutieres syndrome. But you would like to see whether they are efficient enough, and therefore you would like to have a good testing method for your interferon signature, so your interferon activity in the body. Because if that's still high with a medication, you might think of intensifying the treatment. So this is, I think, a very practical talk, and we of course hope that we will have better assays for the interferon production. Any questions? No? Okay. The next talk was about myelin lipid composition in adrenoleukodystrophy, and it was given by another Weinstock from the Kennedy Krieger Institute in Baltimore, and they purified myelin from mouse tissue and human brain tissue from mice with ALD, patients with ALD, and controls, and looked at a lot of lipids, and what they found was that patient myelin of patients who had the cerebral form of adrenoleukodystrophy contained high levels of the C26 LPC, or lysophosphatidylcholine LPC, which is also the blood biomarker for adrenoleukodystrophy. So that in itself is perhaps not that surprising, but what's also the case is that this substance is a very potent driver for demyelination and inflammation. So if you want a mouse, for example, to develop a disease resembling multiple sclerosis, you inject that mouse with LPC. So the possibility is that this high LPC in human brain tissue is actually triggering this neuroinflammatory cascade leading to the cerebral form of ALD. What's also interesting is the mouse model is not a good mouse model for the cerebral form of ALD, and that might be because mice don't have so much of the C26 LPC. You see that even in the genetically modified mouse model for ALD, and even healthy humans have more of that lyso-PC, 26 lyso-PC, than the knockout mice. So this is an interesting finding, and perhaps it helps us to get better animal models for this leukodystrophy. Questions? No? Okay. The next talk was also about ALD, but about female patients who develop, in many cases, an AMN form. And the finding, patients are evaluated and get physiotherapy and advice to exercise at home, and I think you all understand how difficult that is if you have to do that on your own, and even if you have some nudges, it might be a problem. But we also know that directed exercise is really improving your strength and your balance. So this is something where we can actually have good success with relatively modest means, without a fancy gene therapy or small molecule treating these patients. And they looked at remote training in a pilot group with targeted strength training or small group adapted Tai Chi, which was apparently a very attractive program to participate in. And this improves a range of functional measures, including balance. Of course, this was a very small study, so more data are needed, but this might be an attractive rehabilitation program for females with ALD. Any questions for this part? Don't see something. Okay. And then the last talk was about cerebrotendinous xanthomatosis, and it looked at the current available small molecule treatment, but also looked into the future and then at gene-based therapies. And we all know that there is a small molecule for patients with CTX, which is called CDCA, chenodesoxycholic acid, and that's an effective and safe treatment. But for the FDA approval, there needed to be a study, but how do you do a study in patients who already receive a treatment where you know it works and where you know it's safe because you've been giving it to patients for, I think, more than 40 years now. So they did that a trial called Restore trial, where they actually grouped those patients in 2 groups, and every patient got in a double-blind phase either the drug for 2 4-week blocks or placebo, and no one knew when the placebo was given, in which block, and that actually demonstrated what we already knew, that it's, first of all, it's well tolerated, the side effects are there in some patients, many diarrhea and some headaches, but it's otherwise well tolerated, and if you stop it, when you stop it, you see that your metabolic markers, especially the cholesterol, increase in urine and blood. So that was, I think, fortunately enough proof for the FDA, and the treatment is now officially registered, FDA approved, and it's actually a good treatment. You have to take it every day. There are some side effects, but you can do something for this disease. There will be some research into gene therapy with all the possible side effects gene therapy can have. It works in the mouse quite well, so there is a clinical trial in the works, either gene replacement or in the future perhaps direct genome editing, although that's really in the future. These treatments, gene therapies, will be for sure very expensive. This treatment was not expensive, I have to say, and yeah, I think you all know why it's so expensive now, but at least there is a treatment. Any questions for that part? No? Okay. So the next slide is about the posters. There were a lot of young researchers who presented posters on their research projects. We had 3 people who won a poster prize: Carlos Domingos Gonzalez, Guus Forst, and Victoria Kretsching. And I think it's so important to have these young people doing the work which is needed also in the next 40 years when we are old and retired. So this is a nice slide to stop with. So thank you very much for your attention.